cardiomyocytes in the context of their embryonic origin is not well known. It has not been defined. And there are certain questions that have not been well studied, and that's the focus of some of our work. And we're interested in the stepwise mechanism by which progenitors generate mature myocytes in vivo. And the time frame that regulates the transition of progenitors to mature cardiomyocytes, and we are interested in determining whether this is an abrupt change or if it's a progression in this transition. The presence of progenitors in the neonatal period is of interest to us, and also the role of cardiomyocytes and progenitors after myocardial injury. Whether there's presence of cardiac progenitors even during neonatal injury that contributes to regeneration, or is it the responsibility of cardiomyocytes to undergo division? So there's fundamental differences at the transcriptional level between cardiomyocytes generated from pluripotent stem cells and their endogenous counterparts, and that's the area that we're interested in. If we look at the cardiovascular lineage map, both during development in vivo and also as a model from human pluripotent stem cells, there is presence of a mesoderm that gives rise to this cardiac committed mesoderm, which several studies have suggested that this is a or multipotent progenitor that has the ability to give rise to multiple different lineages, including cardiomyocytes, endothelial cells, vascular smooth muscle cells, and fibroblasts. So we've been interested in determining what are these stepwise transitions that a progenitor gives rise to cardiomyocytes. And in terms of the regenerative potential of the human heart, there was a really interesting study that was done in 2009 by Reza and his co-workers, that they did the date stamping of cardiomyocytes based on carbon-14 incorporation into human hearts. And they showed that there was about 10% turnover at the age of 25 and less than 0.5% at the age of 75. What we were interested in was to determine how we can develop an animal model that can directly identify cardiomyocyte division. Up to previously, most of the studies relied on indirect surrogates for cardiomyocyte division, including EDU and BRDU. We were interested in identifying the difference between a karyokinesis and cytokinesis in cardiomyocytes. So we used the MADAM mouse model, which is the mosaic analysis with double markers. And for those who are not interested, this is, or who are not familiar with this, it's the GFP gene that has been cut in half. The C terminus and N terminus are on the homologous chromosome, and the RFP gene are on the C terminus and N terminus on the other chromosome. And homologous recombination occurs at a very, very low rate. And when there is homologous recombination with division, there is generation of a green and a red myocyte. And the fact that this happens at very low rate would actually work in our favor, because we're interested in performing clonal analysis, and we're interested in following individual cells at a very low rate. And we showed that there was, this is an example of a cardiomyocyte, that a progenitor that underwent division generating two cardiomyocytes, red and green. And we showed that there is absolutely no evidence of robust cardiomyocyte division in adults. We did see that generation of clones of cells during cardiac development. And very interestingly, we looked into the presence of c-KIT positive cells in these hearts, and we were unable to identify any c-KIT positive cells that gave rise to cardiomyocytes. All the c-KIT positive cells that we saw, very, very low numbers, were hematopoietic origin. So in order to validate these studies, we developed another mouse model that we called Rainbow by putting 4 different fluorescent genes into the ROSA 26, separated by mutated FLOXP sites, and when crossed with an inducible Cree, we would get expression of one of the markers. And this is a very powerful tool for clonal analysis, because the recombination into one color, even the presence of about 7 cells, the chances of this being a random recombination to that same fluorescent gene is less than 0.005%, making it a very powerful tool for clonal analysis. When we crossed this rainbow mouse with straight Cres with MESP1, NKX2.5, and alpha MHC, we saw the presence of clone formation. And here are a bunch of cells with the color blue and red. However, in alpha MHC, we saw a mosaic presentation of colors. But bear in mind that these are all straight Cres, so the chances of random recombination to the same color is very high. So what we decided to do was take use of the inducible Cre, and we titrated the amount of tamoxifen so we would be able to induce recombination in less than a dozen cells in the entire embryo. And we were able to do analysis after inducing recombination at E12.5. We used beta-actin Cre-ER, NKX2.5 Cre-ER, and alpha MHC Cre-ER. We used beta-actin so that we would be able to induce recombination in all different cell types, including myocytes and progenitors. And we saw that at E12.5, we saw clones of cells, both in NKX2.5, but the majority of the alpha MHC cells that we saw were unable to form large clones. So based on that, we had a hypothesis that cardiovascular progenitor stem cells contribute to heart development, and once cardiomyocytes are formed, they lose their proliferative capacity. And we actually used the rainbow model to prove that this hypothesis is wrong. What we did next was, can you play the video too? What we did next, with the help of Dr. Shai at UCLA, we were able to clear the hearts. We did clonal volume determination. So rather than doing 2D cell counting and using mathematical models to come up with 3D volumes, we actually calculated the entire volume of the cell clones, and we showed that there is a significant decrease in alpha MHC clones compared to the other two. And this told us that there is a difference, at least at E12.5. So we went ahead and did the same experiment, but we did this at E9.5. So we induced recombination at E9.5, and we saw clones again with beta-actin and NKX2.5. But interestingly, we saw that there was formation of smaller clones with alpha MHC. This suggested that perhaps the heart development is regulated by a transition of stem progenitor cells to mature cardiomyocytes, where proliferation gradually declines. So this is more of a transition rather than an abrupt change from progenitor state to adult myocytes. However, it's very interesting that there is a difference from E9.5 to E12.5, so something actually happens between that time point. So we went ahead and looked at the Brdu incorporation in cardiomyocytes from E9.5 and E12.5. You can see that there is a significant difference. And when we looked at alpha MHC expression, it's significantly more in P1, but there's not much difference between E9.5 and E12.5. So we did another experiment where we generated a double transgenic mouse model with alpha MHC GFP and NKX2.5 TD tomato. We took the double positives, and we looked at the Brdu incorporation in these populations. And we saw that E9.5, there is 21% of these cells that are Brdu positive compared to less than 3% at E12.5. This suggests that there is actually something going on between these two time points, that the proliferative capacity of alpha MHC expressing cells go down. So we went ahead and performed single-cell RNA sequencing of these cells at the different time points. And when we did PCA analysis for the E9.5, E12.5, and P1, we were able to separate several clusters. And very interestingly, we repeatedly saw the presence of this P1 outlier that was closer to E9.5 than P1. And when we looked at the gene expression, the E9.5, the P1 outlier was closer in terms of its gene expression to the cardiomyocytes that were at E9.5, suggesting that even at P1, there is probably presence of cardiomyocytes that are developmentally not developmentally closer to the progenitors. So the whole concept of cardiomyocyte regeneration during neonatal period may not be equal among all cardiomyocytes. There may be a subpopulation of cardiomyocytes even during the first week that have the ability to undergo division. To test that hypothesis, we actually performed an LAD ligation in neonates and in adults. And of course, we showed the presence of clones in neonates, indicating that these cardiomyocytes are undergoing division, where there was lack of clonal formation in adults. And we believe that there is a subpopulation of cardiomyocytes during the neonatal period that have this ability to undergo division. So in terms of the cardiovascular development, traditionally there has been a thought that there is a clonal model where there are progenitors that have the ability to expand. There is a hierarchical model that there are stem cells that have the ability to give rise to transiently amplifying cells. However, our model suggests that there is a clonal hierarchical model in cardiovascular development where are different stem cells at different stages of development, and these have the ability to give rise to transiently amplifying cells. And in adult hearts, the very, very low rate of cardiomyocyte division is actually due to symmetric division. So in terms of cardiac regeneration, considering the hostile environment post-injury, many studies have shown that bone marrow cells don't work, skeletal myoblasts, they have long-term engraftment problems. Endogenous cardiomyocytes and resident cardiac stem cells, we have shown that they are probably not a source for cardiac regeneration. And we have placed our attention on embryonic stem cells as a method for cardiovascular development and injury. So our approach has been to generate cardiac-specific reporter lines. And we have generated several reporter lines, including NKX 2.5, ISL1, TBX5, and alpha-MHC. And what we do, we prospectively isolate these cells and characterize them both in vitro and in vivo. One of the lines that we've generated is TBX5, and this is by making a line by using CRISPR-Cas9 technology to incorporate the gene into the TBX5 locus without disrupting the gene, so it's not haploinsufficient. And as you can see, by day 12, we get a large population of these cells. And we transplanted them into the mouse heart, and we showed that they survive, but they do not engraft, they do not integrate, and they show very immature structure. And we went ahead and generated multi-reporter lines by using MLC2V and also troponin. And very interestingly, to our surprise, we found that even the TBX5 negative cells can generate cardiomyocytes. And we performed RNA sequencing on all of these populations, and we saw that the TBX5 negative populations that generate beating cardiomyocytes are enriched for second heart field genes. So our hypothesis was TBX5 may be a good marker for first heart field progenitors, whereas NKX 2.5 is expressed on all cardiac progenitors. So taking the double positive, those cells that express NKX 2.5 and TBX5, we can separate first heart field from second heart field that would be NKX 2.5 but not expressing TBX5. And we were able to optimize our differentiation so we can get a large population of double positive. We performed single cell patch clamping, and we looked at the propagation of unstimulated spontaneous generation of electrical activity. We looked at their exposure to pharmacological agents, and we looked at their APD90 histogram showing that there are two peaks suggesting of two different types of myocytes. We did the same thing for a second heart field. These are the NKX 2.5 positive cells that don't express any TBX5. And very interestingly, we were able to show that there is a third population that are only TBX5 expressing cells, not expressing any NKX 2.5. And when we characterized these cells, we showed that these are the cells that are responsible for generation of nodal cells. We went ahead and compared those to fetal hearts. These are human fetal hearts. And we showed that these cells, the first heart field, are closer to the left ventricle compared to the right ventricle. And we performed single-cell RNA sequencing of these two populations, and to our surprise, we saw that there are two lobes that are completely different, indicating that the first and second heart field, besides their anatomical location, they also have different gene expression profile. And we showed that they're enriched for cardiomyocytes, both for ventricular and atrial cells, and they don't express any, there's no evidence of any nodal cell expression. As you can see, ISL1 is highly expressed in the second heart field. And we also looked at our gene expression analysis to identify any cell surface markers, and we identified corin as a very efficient marker to separate the primary heart field progenitors from the second heart field, and we showed that this is also present in adult cardiomyocytes and by gene expression comparing them to fetal heart as well. So we have started doing some large animal studies where we transplant these cells into pigs after undergoing myocardial infarction, and we followed them by MRI, tracking the transplanted cells. And when we look at the expression of cardiac-specific markers, we see well-organized ultrastructural protein formation. However, unlike other reports, we have not been able to see any connexin 43 gap junction between the transplanted cells and the host. We see them between the transplanted cells. So this is, can you play the videos, both of them? So I think the ability to generate first and second heart field and LV and RA, this is not showing very well here, but the propagation goes from one side to the other, and we put a probe here, and we can actually take the electrical propagation from one side to the other. And this is a spiral formation, indicating that these cells are these are very pure cardiomyocytes and have the ability to perform well for not only disease modeling, but also for drug screening, and most importantly for regenerative medicine, because we hypothesize that perhaps one of the reasons for seeing arrhythmia in transplantation of the human embryonic stem cells or pluripotent stem cells is probably the heterogeneity of the cells that have been transplanted, including some nodal cells. So in light of all the recent controversies regarding stem cell therapy for regenerative medicine, I think that we have not been doing the research in a way of setting forth a hypothesis and doing our diligent work to prove the hypothesis wrong. And this is reminding me of a story that 3 umpires were asked how they call balls and strikes. One said that I call the way I see them, and this person acknowledges perception and reality are blurred. The second said, I call them the way they are, and this person confuses perception. And the third said, they ain't nothing till I call them, and this person defines reality. And that's the case that we've seen with a lot of stem cell research, that we set our mind about a finding and we do our best to prove that rather than to disprove it. So I'd like to thank my laboratory sources of funding and also my collaborators. And also this is a picture of my son, who's taken a keen interest in medical research, but he falls asleep pretty fast after being exposed to it. Thank you very much.
Thank you for a very nice presentation. We are a little bit behind, so we have time for one quick question.
So, Reza, this is a hardcore molecular cellular biology. With all your tools, rainbow mice, and the knowledge, the fundamental question is why the postnatal day 1 to day 7, the cell exits, myocyte exits cell cycle. If you can speculate what's going on, the myocyte in the fetal, they divide.
Yeah, so I think how I would speculate on that is that the myocytes during the neonatal period are not all at the same developmental stage. There are some that are more mature than the others, and we think that they don't all have the ability to undergo division, and we think the more immature cells the more immature cardiomyocytes have the ability to undergo division. And the goal is to identify those and determine what causes them to have this high ability of proliferation and the cell cycle genes that are enriched in them.
I guess we need to identify the heterogeneity of the cell type, but also probably want to identify the regulators that after birth, postnatal day 1, day 2, they all exit cell cycle. If we know these regulators, then we can manipulate the regulator, we make the regeneration.
I completely agree with you, and I don't think that it's one factor. I think there are so many different factors that play a role, and that is one of the things that we've become interested in is the regeneration of the neonatal heart cannot be only defined by cardiomyocyte proliferation. It has to be accompanied by prevention of fibrosis. And the cardiac fibroblast, and specifically epicardium, we believe plays an important role, as it has been shown in zebrafish, that the epicardium gets activated and sends signals for cardiomyocytes to undergo division. And we see that during neonatal period, and that is blunted in adult hearts.
All right, thank you for a nice presentation.